So first, let's create the Cas9. We will need to create 3 ellipses. 1, 2, 3. When I'm duplicating it, I was holding Alt or Option on Mac, and then drag the shape. Okay, and very quickly just manipulate the shape of these ellipse. Okay, this one needs to be slightly bigger, and this one align here. Okay, good. And then I will select, this one should be even higher. Okay, good. Once you're happy with them, select them all and go to Shape, Format, and here you'll see Merge Shape, and use Union to combine them all. Okay, and now we need to give it a gradient. So let's go to our, I was going a little bit too quick. Let's go back. So now we need to give it a gradient. And to get the gradient, we will need to come here, Shape Fill, and then to go to Gradient, More Gradients. And this shape, Format Shape panel will open up because I already have it here, so you didn't see it pop up. However, you should be able to find the Gradient Fill here. So by default, I think you will be seeing something like this. That means it's a linear gradient, and we don't want that. We want a radial gradient, so make sure you come here to switch the type to radial. And in direction, it needs to radiating from the center. And then you can get a more focused volume. And then we only need 2 color boxes, so make sure you delete the redundant ones. Remember to move your white color box just slightly into the gradient stops. Somehow if you put a little bit to the right, and the white gets really focused. That's what we want. As you can see in the example, the bright light is very focused. Okay, now the gradient is ready. We will give it a gray outline, and we need a thinner one, so let's go to the half point line thickness. Okay, that's good. I will make it a little bit smaller. All right, now we're ready to create our target DNA. So first, let's grab our free shape tool, free form. This is very important because free form allow us to edit points, edit the anchor points in the shape. So we want this to be our DNA line. So first click on the canvas and then hold Shift to keep the line straight, and then make the second stop. Once you have that, and then press Enter to make it into a shape, which is the line here. And then we will increase the thickness, and let's use 2¼ thick, and then we will need to give it a round cap so it is more organic, and then the line color, the stroke color is the gray. All right, and how do we create this sigmoid transition? So let's go back to the shapes, and then here you will find a curved connector. So this will be automatically create the sigmoid shape for you. And another really nice thing about this shape is that PowerPoint will help you to directly snap it onto your freeform shape. So it will be perfectly connected to your DNA line. So you can see that the sigmoid shape is gone, but if you just move this anchor point a little bit away from the other one, essentially making the sigmoid, the curve a little bit bigger, then you will get the sigmoid shape back, and then we can scale it down then, like that. Okay, good. We will move them here a little bit for now, and I will keep pushing it in. Great. All right, and this one we will use the same style as our straight line. Okay, now we have this ready, then we can duplicate it, and then that's the other side of our shape. And I will try to flip it horizontally here. Do it manually like that. Okay, good. And now we will give it a red color instead. This one. Good, and I will move it in, because as you see in the example, half of the PAM is in our Cas9. And then this one, the same. All right, and now we will add in the proto spacers. And so you can just copy the DNA line you already have, and then place it on top, and then let's make it pitch black. Then you have your proto spacer. Let's group these together, and then I'll make a copy of it. We'll do the rotation instead. Okay, good. Here, distance is about here, and we need to change the color of our PAM here. So go to the copy style and then change that. And this one needs to be red. Okay, good. Let me zoom in to take a look. Yeah. So they should be snapped perfectly, and if you see it a little bit crooked, that's just because of the zoom view of your PowerPoint. So if you zoom in a little bit, then you'll see they're just fine. Okay, now we need to create the base pairs. So for the base pairs, that's use the line tool to just very quickly create these line, and we don't want it to be completely snapped to our DNA line, so make sure there's some space in between, and make it gray. Then we can start duplicating it. It's okay that they're not properly aligned now, because we can just use arrange to help us to align everything in the middle, and also distribute horizontally. Oh, okay. Then I'll group this. And then I'll make a copy so we have it on the other side as well. That's good. Okay, now we will create our CRISPR DNA. So let's copy the DNA line and then place one underneath our proto spacer. And then just elongate it. Oops, that was a little bit too long. Okay, good. Over here. Yeah, I think I will move these a little bit closer like that. Okay. All right, so if you copy these base pairs, then you will have the same, then you can place it over here, so you don't need to recreate it again. They are way more dense than mine, so I will quickly fix this. I'll speed up this part. Okay, so we will put on the CRISPR RNA, which we will use a rectangle. Fill that is yellow, and the outline, also the thickness, half points. Okay, let's put it here. Like that. Cool. Okay. Now we can start labeling. Oh, this one should be black. Let me quickly change it to black. Okay, we can start labeling. So we will use our text box to create some text. That's 3'. And the how to call that? The font is Helvetica. And then we use 10 as the font size. And then we can put it here. Ooh, I'm copying the wrong thing. And we will start copying these text boxes. Let me see. Good. And here we have it at 5'. Okay, so for the scissors, there are free icons that you can just download from the internet, but as a scientific illustrator, I will very quickly show you how to create the scissors. So let's use this donut shape to draw the hand holding part, make it pitch black, make it smaller, a little bit thinner. Right, and then this is not entirely a graphical abstract thing, so yeah, just very quickly do it here. Okay, then edit points. This anchor point, I hold shift to make it smooth, so it will be a round shape. And now I can use it as the blade. So the blade should be... Let's group all of these. And then we can create the two types of CRISPRs, Cas9. First one, we will be creating this structure called the trans-activating CRISPR RNA. So let's get into it by first let's create a circle, and then a rectangle. Put it this way, and let's go to arrange to make it sure that it is centered and aligned. Good. Once it's done, let's go to merge shape and click on union to unite all of them. Let's give the outline the red that we want and cancel out the fill. The thickness of the line needs to be 2.5 points. All right. Then, so how do we create these two parts? So let's go to edit points and then select this anchor point, right click on it, open path. So now you break the connection between these points so you can swing it to the other side. All right. Okay, and then the part that will be connecting with our CRISPR RNA, we will go to edit points, and then we'll go to edit points and right click, add point, and then this one we can swing it all the way here and then drag the handle downward to here, and this one, let's elongate it a little bit, move it downward like that, so they will be a good site. And let's turn it to let's give it some rotations and then adjust the size so we can move it to their complementary sites over here. Okay, the double strand part, the pin part is outside of Cas9. That's good, and let's go back to our edit point. Then we can put it here. Okay, and then how do we create this one? The CRISPR trans-activating CRISPR RNA chimeric. So that's first copy the DNA line from here. And let's first create the linker loop. So let's first create the linker loop we can come here to grab this connecting curve. So let's connect this one to the DNA line. I cannot reach it because of this 3' end. So let's do it again. I connected it here. That's good. Okay, so I will connect it by I'll connect it by using a curved connector like that, and then connect it to here. It looks a little bit strange, but once you drag on this yellow box and then move it all the way here, then it looks just fine. Okay, and if you give it a purple color and the thickness that's the same of the DNA and the RNA, then you will see it become the linker loop. And now we only need to switch this part a little bit to the shape. So first make it red. Okay, it acts a little bit strange when it's connected. So first I will remove it. And then select the red RNA line. And then first I will enlarge it a little bit. And edit points. And when you right click on it, you can add points. So that's how you draw the CRISPR-Cas complex. And so now you know how to draw a Nobel Prize winning research. So I will make another video showing how to make the kind of illustration they use in the Nobel Prize press conference in their PowerPoint. I will be using Illustrator in that one, so make sure to subscribe to DrawBioMed so you don't miss the tutorial. And I wish you all happy DrawBioMed, and I look forward to seeing you in the future videos.